Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture by Biology. In the series of video lecture, we are going to talk about recombinant DNA technology in detail. Okay, this is the first video lecture regarding to recombinant DNA technology, and in this first video lecture, we are going to talk about the overview, the concept, the big picture of, or the summary of recombinant DNA technology. So let me write. In short form, we can write that RDT, R, D, and T. R stand for recombinant, D stand for DNA, and T stand for technology. Recombinant DNA technology. We also call that gene cloning. We also call it gene or DNA cloning. DNA cloning. Now, what is recombinant DNA technology? Recombinant DNA technology is the technology, or it is a technique through which we can join two DNA molecules from different sources. We join two DNA molecules from different sources of the organism, and we get a desired genes from these joining DNA molecules. Okay, we call that recombinant DNA technology. Why we call it recombinant? Because in this case the DNA are recombined to each other. And now, what are the DNA or gene cloning? DNA or gene cloning, before that we need to understand that what are actually cloning. Cloning means to produce the same copy of the things which we need. For example, if I have this marker, so how I will produce the same copy of that marker? This is called cloning. Okay, in this case we use what does DNA cloning means? DNA cloning means we should produce the same copy of the DNA, or we should produce the same copy of the gene. Okay, for example, we have the human genome, and inside the human genome we have the insulin. Now from insulin, let's suppose in this case, let me take this is, let's suppose, the DNA. Okay, human genome or DNA. At that specific position we have the genes. Let's say this red one representing the genes. Okay, now this is called, first of all let me tell you about the genes, difference between the genes and DNA. DNA are the hereditary material that can store the genetic information, or that can store the information, and genes are the specific area of the DNA which have the ability to produce the protein or the product. Okay, so these are the genes. Now when we produce the same copy of the genes again and again, we call that cloning technique, gene cloning. And when we produce the same copy of the DNA again and again, we call that DNA cloning. Okay, so that's a little bit about the introduction to recombinant DNA or gene cloning or DNA cloning. Now, what we need in recombinant DNA technology? We need different kinds of things or molecules or substances in recombinant DNA technology. Let me write each of the molecules. We need target DNA, or genome. We also need enzymes. In this case, we need restriction enzymes, ligase and polymerase. So let me write, we need ligase, we need restriction enzyme, restriction enzyme. We need polymerase. Okay, these kinds of enzymes we need in recombinant DNA technology. Moreover, we need a vector. Different kinds of vector we need: phasmid, plasmid. We need plasmid. These kinds of vector we will be talking in the next video lecture. And we also need host cell. Usually in recombinant DNA technology we need a bacterial cell. Okay, what we need a bacterial cell, and usually we take E. coli. So that's the things, that's the material, that's the substance, that's the molecule that we need in recombinant DNA technology. Now then, what kind of steps involved in recombinant DNA technology? There are different steps involved in the recombinant DNA technology, but the main step through which the recombinant DNA technology process are performed, these are, let me write, isolation of the target DNA, isolation of the target genes. What kind of genes we need, we should isolate these kinds of genes. This is the first steps. The second step is cutting of target genes, or DNA. Okay, cutting of target genes or DNA by using restriction enzymes. And we also need joining. They also contain another step, we call it joining. Joining of DNA. And then amplification of DNA. Amplification of DNA. Okay. Now there are some other steps also, but these are the main steps that are usually involved in recombinant DNA technology. Now what actually happen in isolation steps? In isolation steps, we should select a cell which have our desired genes. Let's suppose we take the human cells, they have the desired means genomes, or they have the desired DNA which contain the desired genes to produce insulin. We should select that kind of cells. Now once we select the cell, then how we isolate that? We should treat these cells with the help of detergent, due to which the cell membrane is ruptured and the genome is or the whole genomes is released out. Okay, this is the first steps. The second step is the cutting or ligation, cutting of the target genome. We should cut that genome what we need. Okay, now let me explain each of the step with the help of diagram. What actually happened? Now what actually happened? Let's suppose we should select a cell. Okay, now this is, let's say, a cell we have of human cells. Okay, inside the human cells there are the genomes, they contain the genomes present inside the human cells. Okay, and inside the genomes there is also a target DNA portion or a target genome portion. They have, let's say, this red one represents the genome or the target genes which we need. Now this target gene, let's say in this case, these target genes have the ability to produce insulin, which is a product which are especially used in the diabetes disease. Okay, so let's say this is the insulin. Okay, now how we isolate this genome? We should treat the cell with a detergent. We should treat this cell with detergent. Due to this way, the cell membrane is ruptured. All the material, all the content inside the cell will released out. And finally, we also get our gene of interest, or not gene of interest, we just get the whole genome of the human. So this is, let's say, is the whole genome of the human. Okay, and they also carry our desired genes, or we can say insulin genes. Okay, this is the first steps. Now we isolate the genes. The second steps, we should cut their genes, or we should cut the whole genome with an enzymes. And in this case, we use restriction enzyme. So we should cut this genome with restriction enzyme. Okay. Let me a little bit explain the restriction enzyme, because it is very important in the molecular biology, or it is very important in the recombinant DNA technology. We also call it restriction enzyme, or we call it restriction endonuclease. Okay, let's say we have sequences in this case. Now to understand the concept of restriction enzyme, let me tell you that we have a sequences in our DNA. Let me write E, A, T, T, and C. Okay, and we have the same sequences are present under the this sequence. We have G, A, T, T, and C. Okay. Now let's say these are the sequences of the bacterial genome, or these are the sequences inside the DNA. Okay. Now these sequences are called as palindromic sequences. We call these sequences palindromic sequences. Palindromic sequence. Now why we call it palindromic sequences? Because in this case the nucleotides which are present up at the upper strand, the same nucleotide is also present at the lower strand. Okay, but it is opposite to each other. Okay, as you are watching GAATTC and again GAATTC. Now both the sequences are similar to each other, but they are just opposite to each other. Okay, both are the sequences, but they are actually opposite to each other, and we call that palindromic. They are palindromic to each other. Okay, that's why we call it palindromic sequences. Okay, now here we use the restriction enzyme to cleave. Let's say these genomes have the ability, or these are the target genomes that can produce the insulin genes. Let's say in this case, now how we cleave that? We use the restriction enzyme. Now why we use the restriction enzyme? A restriction enzyme we use for that purpose because restriction enzyme can cleave the DNA, the target DNA at a specific points. At that specific points, they can cleave the DNA. They can cleave the DNA just like that pattern. Okay. Now the first thing. The second thing is that we also call it restriction endonuclease. Why we call that restriction endonuclease? We also call that restriction endonuclease because they can cleave the DNA at the midpoint, as you are watching. The DNA is cleaved at the midpoint. Due to this way, we get a sticky end. Let's say. Now this is the DNA that is cleaved. We get just like that pattern, that fashion of the DNA. Now this is, as you are watching, these are the sticky ends which are produced, sticky ends. Okay, so that's why we call that restriction enzyme, because they can cleave the DNA at a specific point. They can cleave the DNA at the mid position. Okay, we need this is restriction enzyme. Okay, in this case, there are many types of the restriction enzyme. Let's say we take EcoR1 in this case. Although there are also BAMH1 are also, but let's take EcoR1 enzyme, EcoR1 restriction endonuclease or restriction enzyme. Now they can cleave the DNA at just like that pattern or that patient. Then after that, what actually happen? We need a vector. We also need another things. We need a vector. So let me draw a vector here. So this is, let's say, a vector. All of you better know the vector is a circular in shape. So this is a vector. It is a circular DNA, or let's say in this case we take the vector plasmid. Okay, this is a vector plasmid or phasmid or cosmid. These all are called vectors. Now what are the vectors? We take here, this is the vector. Vector are the delivery system, or they are delivery material, or they are the transfer material. They can transfer our target genes, our target genome. Okay, just like plasmid. Plasmid is also a vector because they can transfer our target genes. They are a delivery system. They are a vehicle through which we can transfer our genome, our genes of interest. Okay, so we need a vector. In this case, usually we need the plasmid. Although we also need phasmid, cosmid, these also vector we also need. But let's say in this case we need a plasmid vector. Now, after that, what actually happen? Now we treat this one human genome with the EcoR1 or with restriction enzyme. Once we cleave this genome with the restriction enzyme, then we get our desired genes. Let's say this is our desired genes we get with the sticky ends. Remember, the same enzyme, we also apply the same enzyme with this vector. Now why? Why we not use different kinds of enzyme? We use this one enzyme with we use this one genome with the BAMH1, and let's say we use this one vector with the EcoR1. Why we not use different kinds of enzyme? At that case, we use the same enzyme because we should produce the complementary end at that position and at that genome. And because if we use the same enzyme, so we get the same complementary end at that position and as well as at the vector also. If we use, let's suppose if we use BAMH1 for this one genome and we use EcoR1 for this genome, so we get the sticky end, but that sticky end are not similar to each other. They are not complementary to each other. Due to this way, the target genome is not bind to this vector. The target genome are not bind. Why? Because the enzyme are different and the complementary end or the stick end which is produced, that is also different to each other. Due to this way, the vector will uptake the target genome. Now let's say this is our target genome are inserted. Our target genome is now inserted into the vector. So this is the target. Now once we insert or inject our target genome, then still it is not completely linked. As let's say this is now let's say the vector are just bind here, but it is not sealed. As you are watching, the vector are not sealed with each other. Now at that case, for the ligation of the vector to the target genome, we will provide another enzyme. We call that ligase enzyme. Then at that case, we will provide the ligase enzyme. With the help of ligase enzyme, they will seal the nick or the space which are present between them. And due to this way, the genome are now completed, or the vector is now completed with our target genome. Once the vector is completed with the target genome, then what we should do? We should transfer this target, or now this is called recombinant DNA, or we also call it recombinant DNA plasmid. Why? Because they can contain the DNA which the desired DNA or the desired genome. Okay, that's why we call it recombinant DNA, because we recombine the vector with our desired genes. That's why we call that recombinant DNA or recombinant plasmid. Now after that, what we should do? We should transfer this recombinant DNA to a host cell, and in this case, as I told you, we use a bacterial cell. In this case, what we use? We use a bacterial cell, and usually we use a E. coli cell. So let's say this is the E. coli. So once we get that E. coli, then how we will transfer this plasmid, recombinant plasmid, to this E. coli cell? We should use a transformation process. Okay, so next step is the transformation. Now how the transformation are take place in a bacterial cell? We should treat this bacteria with a calcium chloride, or we should treat the bacteria or we provide the electric current to bacteria, not high potency electric or not high electricity. We just provide a little bit electric current to the bacterial cell, or we treat the bacterial cell with the calcium chloride. Due to this way, so after that, the bacterial cell can forcefully take the target genome, or they will get the vector. Okay, once they take the recombinant DNA plasmid or vector, then what actually happen? Now this E. coli cell can contain our plasmid. There is a lot of E. coli are present, and this is not only one vector, and this is not only one genome, and this is not only one target genome. there are different kinds of human genomes, different kinds of human cells. We use detergent with with the cells on the human cells, and then we get different a lot of human genomes, and then we use a lot of, so we not use only one means enzyme and one cell. No, we use a lot of enzymes, the same enzyme a lot in a lot number, the same cell in a lot number, the same genome in a lot number, the same vector in a lot number. Okay, due to this way, there is also a lot of cells, E. coli cells, or different kinds of cells we have. Not only the E. coli, we have different kinds of cells. But when we put calcium chloride or when we provide electric current on each of the cell, then what actually happen? There are some bacteria that can uptake the recombinant DNA plasmid. Okay, not all the cells will take. Okay, now once they get the recombinant DNA, so then what actually happen after that? We should, grow each of the cell with agar plate. Let's say this is the agar plate. This agar plate contain ampicillin antibiotic. Now due to this ampicillin antibiotic, those bacteria which don't take the vector or the plasmid, such bacteria will die, and those bacteria which take the plasmid, they will survive and divide it. So this is called screening. Okay, so we get our desired bacteria. So let's say these are the this colony shows that each bacteria contain the plasmid, recombinant plasmid, and each bacteria are survive. Okay, now after that we take each colony and we will get the desired genes again and again. Okay, now this is called, we get a lot of petri plates, and these a lot of petri plates contain the same bacteria, E. coli, which can carry the desired genes or insulins. Then after sometimes there are the genes can produce insulin proteins or insulin product. Okay, why? Because inside the bacterial cell the transcription translation process is going on at a time. So although there is also in the genes of the bacterial cell is also present, the genome of the bacterial cell is also present in E. coli as well. Now whenever this genome is replicated, definitely this plasmid is also replicated, because plasmid is self-replicating. This is a self-replicating genome. Okay, now whenever they whenever the polymerase enzyme which need for the bacterial genome, so the same polymerase enzyme is also needed in this case. Although there is no enzyme is used for the plasmid to replicate, they are self-replicated. Okay, so whenever the genomes are replicated, so the bacteria is divided into two, and then we get 4, 8, a lot of bacterial cell we get. Okay, then we get a colony which is visible to our eyes. Okay, then we get our desired genes, and we get from this desired genes, finally we get the our product, means insulin. Now let me explain the whole process from the beginning, that what actually happened. First of all, what we have, we discussed that recombinant DNA technology. It is that process that in which we can join two DNA molecule with each other. Okay, that's why we call it recombinant DNA. Now then we talk that what are the gene cloning and DNA cloning. The gene cloning and DNA cloning are that process in which we get the same copy of the DNA or the same copy of the genes. Okay, we call that gene cloning or DNA cloning. Then we talk that there are different material or different substance or molecule which we need in recombinant DNA technology. We need a target DNA or genomes which can contain our desired genes. Let's suppose in this case this is the DNA, and we need this one DNA. Why? Because they can carry our desired genes. Let's say in this case they can carry the insulin genes. That's why we take this one target DNA, or this is the target genome and this is the target DNA. Okay, then we talk that we need different kinds of enzyme: ligase, restriction enzyme, and polymerase. Then we talk that we need a vector, we need host cell. Okay, then we talk that there are different steps are involved in the recombinant DNA technology. Okay, they contain isolation step, cutting of target genes or DNA, joining of DNA, amplification of DNA. Okay, now here one thing that is very important, that we always use the gene cloning, but PCR is also used for the copy of, to produce the copy of the DNA. Okay, we use gene cloning to produce the same copy of the genome or DNA, but PCR is also used for to producing the same copy of the DNA. Now why we don't use here the PCR? The answer is that we don't use PCR in this case because PCR is an artificial process. If we use PCR in this case, so we just get, let's say we use PCR for this means for this DNA, for this target DNA which contain our desired genes or which contain our desired product. Now if we apply PCR, so what we get the same copy again and again. But remember, we didn't get the protein. For the protein synthesis, we should put this DNA at that genome which contain the replication origin. Because for the transcription translation, we need the replication origin. We need the start codon and stop codon. Due to start and stop codon, due to replication origin, then we get the what we get? We get the desired genes. Now remember, if we use PCR, so we just get the DNA segment again and we get a lot of DNA segment, but we didn't get the protein. Why? Because it doesn't contain replication origin as well as start and stop codon. Okay, so that's very important point, so note it. Now then they contain isolation step, cutting of the target genome, joining of the DNA, and application of the DNA. Now then we explain each of the step with the help of diagram. Now what actually happened, we should select this target cell. Let's say in this case we just select the human cells which have the genome. Now these genomes carry a target genes which have the ability to produce insulin product, okay, or the target DNA which can produce insulin product. Now we should treat this cell with the detergent. Why? Because once we treat the cell with the detergent, then the cell membrane is ruptured and finally we get the genome. Now once we get the genome having the desired genes, then how we isolate this desired gene? The second step is that we should cut the target DNA with a restriction enzyme. Now we apply restriction enzyme with this our whole genome. Once we apply the restriction enzyme, due to this way they will cleave our target genome or our target genes or our target DNA. Now once we get our target DNA or genes, then after that what actually happen, we also need another things we call that vector. We talk that vector are the vehicles or the substance or the vehicle substance or the transfer substance that can carry our genes through the bacterial cell, that can carry the genes to the bacterial cell. Okay, so we call that vector. In this case we take a plasmid vector. Okay, now as we treat the genome with restriction enzyme, let's say we treat the genome with EcoR1. Okay, then we also talk that the same enzyme is also applied on the vector. No, why? Because we talk that if we didn't apply the same enzyme on each of the genes, on each of the vector or on each of our genes, then it will not produce the sticky end. It will not produce the sticky end which is complementary to each other. Okay, that's why if we use EcoR1, if we use the same enzyme for the our own genome as well as for the vector, then it will produce the sticky ends, due to which each of the sticky ends are complementary to each other. Due to this way, the target genome as well as the vector are joined to each other. Okay, they are complementary to each other and they are connected with each other. After that, we need a ligase enzyme. Now what actually ligase enzyme do here? The ligase enzyme will seal or will join the space, or they will fill up the space. Okay, so for that purpose we need the ligase enzyme. Now after that what we get a complete vector, or after that we get a recombinant DNA plasmid. Now why we call that recombinant DNA plasmid? We call that recombinant DNA plasmid because they have the genes of our target. They have the target genes which we need. Okay, and we recombine that target gene with the ligase enzyme. Now after that we should transfer this vector to the bacterial cell. Now how we do that? We should treat the bacterial cell with a calcium chloride, or we should treat the bacterial cell with a electricity. Okay, we call that competency. When the bacterial cell uptake directly or forcefully uptake the vector, we call that competency. So we just influence the competency of the bacterial cell, due to which they can uptake the vector. So there are a lot of bacterial cells are present. So not all bacterial cell will uptake the vector. Some of the bacterial cell will uptake the vector. Now how we know that which bacteria can uptake the cell? For this purpose we should perform the screening. Now how we perform the screening? For screening we should prepare agar plate. And we should prepare those agar plate which contain the ampicillin antibiotic, which we use as a marker. Due to this way, we should grow each of the means. We should pour our solution which contain the bacterial cell, all the bacterial cell and all the plasmid. We should pour that solution on the agar plate containing ampicillin as a marker. Now those bacteria which don't have the plasmid, such bacteria will die, and those bacteria which have the plasmid that contain our desired genes, these bacteria will multiply, and these bacteria will multiply through a binary fusion, and due to this way our target gene is also multiplied. Why? Because translation process is going on in the bacterial cell every time. Okay, due to this way our own plasmid is also replicated. Moreover, this plasmid they have also a self-replicating ability. Okay, now after that, once we get a lot of bacterial cell, so we get a colony. So we take that colony and we should put that colony into a next agar plate. Okay, due to this way the bacterial cell is divided, and finally we get our desired genome. Now how we get the product? Because the bacterial cell is divided, and due to this way our plasmid is also divided, and after some time we get a lot of plasmid that can carry our desired genes, means insulin. Now after some time, once we get the insulin, so then the insulin is produced by this target genome, and we finally get our desired product, and then we use it for the diabetic disease. Okay, so that's how we can perform the recombinant DNA technology for the molecular level. So that's how we can perform the recombinant DNA technology. Now this is the overview, this is the summary, this is the big picture, this is the concept of the recombinant DNA technology. Now once you get the idea about the recombinant DNA technology, so then the rest of the video which are regarded to the recombinant DNA technology will be very easy for you. Okay, inshallah in the next video lecture we are going to talk about vector, okay, in a detail. Okay, so please stay tuned and continue to watch the video. Thank you so much.